So let's take a moment to understand some of the structures, the functions, and the physiology of the red blood cells. That is, these are the erythrocytes. The erythrocytes are the most numerous of the formed elements. The average adult has about 5 million erythrocytes per microliter of blood. This is compared to only a few thousand leukocytes per microliter of blood. The erythrocytes are flexible so that they can bend and fit through small capillaries. The average capillary diameter is about 9 micrometers. However, many are much smaller than the 7.5 micrometer diameter of the erythrocytes. Fortunately, the erythrocytes are flexible. This is due to the flexibility of the structural proteins that help to maintain their shape. So once they encounter a larger vessel, we see that they will spring back to their original shape. Speaking of their shape, they are shaped like a biconcave disc, so that is, their edges are much thicker than the center of the cell, and the center on both sides is indented. This shape helps to give them a much greater surface area compared to their volume, which is ideal for gas exchange. So remember that physiology likes high surface areas for gas exchange. And we see that a sphere of similar size would have a much smaller surface area compared to its volume. So it wouldn't be as well suited for gas exchange as the biconcave disc is. And so this is why the erythrocytes are not shaped like spheres. They have no nucleus and very few organelles. They do have a plasma membrane. And the inside of the erythrocyte is filled with a protein called hemoglobin, which carries oxygen as well as some carbon dioxide. The fact that they don't have many organelles is very beneficial because it also means that those organelles aren't using any of the oxygen that the red blood cell is carrying. They do need to make some ATP energy, but they do so anaerobically. That is, they do so without the need for oxygen. The primary function of the erythrocyte is to transport respiratory gases, primarily oxygen. They do transport some carbon dioxide. However, they do not transport most of the carbon dioxide produced by our body. Instead, what they do is that most of the carbon dioxide is converted into a substance called bicarbonate, which forms the alkaline reserve in the bloodstream. The erythrocytes do function in the conversion of carbon dioxide to bicarbonate. So how does the erythrocyte transport oxygen? It does so using a protein called hemoglobin. So if we ignore the water content of the erythrocyte, roughly 97% of that erythrocyte is hemoglobin. Hemoglobin, which is abbreviated HB, is a large molecule that's made of 4 folded protein chains as well as iron atoms that accompany it. Hemoglobin consists of these 4 red-colored heme groups, and heme is a pigment that gives the erythrocyte its red color. Each heme also has a single iron atom at its center. And we also see that there are 4 globin protein chains that support those heme groups. But each iron atom in those heme groups can bind and carry a single oxygen molecule. So therefore, one hemoglobin can carry up to 4 oxygen molecules in total. So as mentioned previously, each heme pigment molecule has a single iron atom at its center, and that iron atom is what binds and carries the oxygen molecule. So each iron atom can bind and carry 1 oxygen. Therefore, how many oxygen molecules can a single hemoglobin carry? So if your answer to this question was 4 oxygen molecules, you would be correct, because 1 hemoglobin molecule contains 4 heme groups, and so it can carry 4 oxygens. Each erythrocyte has about 250 million hemoglobin molecules packed inside of it. This means that 1 erythrocyte can carry about 1 billion molecules of oxygen, since 250 million hemoglobin molecules multiplied by 4 oxygens equals 1 billion oxygen molecules. So how much hemoglobin on average is found in a 100 milliliter sample of blood? So let's draw 100 milliliters of blood and examine roughly about how much hemoglobin is contained within that, the erythrocytes of those in that 100 milliliter sample of blood. The average for adult females is anywhere between 12 to 16 grams of hemoglobin per 100 milliliters of blood. The average for adult males is anywhere between 13 to 18 grams of hemoglobin per 100 milliliters of blood. And the average for infants is anywhere between 14 to 20 grams of hemoglobin per 100 milliliters of blood. So for scale, 6 pennies weighs about 15 grams. So that is, each penny weighs about 2.5 grams each. So that gives you kind of an idea of roughly how much hemoglobin is in a 100 milliliter sample of blood. So in the lungs, oxygen will bind to hemoglobin. And hemoglobin that is then saturated with oxygen is referred to as oxyhemoglobin. At the tissues, oxygen is unloaded, so that is, it becomes unbound from hemoglobin. Hemoglobin that has unloaded its oxygen is called deoxyhemoglobin. However, we see that hemoglobin almost never unloads all of its bound oxygen molecules. So in this way, the blood always contains a little bit of a reservoir for oxygen. We also see that hemoglobin changes shape when oxygen is loaded or unloaded from it. When oxygen is being loaded in the lungs, hemoglobin changes shape so that it's much easier for the next oxygen molecule to bind to it, and so on. This ensures that the last oxygen to be loaded onto that particular hemoglobin molecule is loaded on very easily, and hemoglobin will generally be very well saturated with oxygen at all times. So similarly, at the tissues, when oxygen is unloaded, hemoglobin changes shape again, and this shape change actually makes it easier for carbon dioxide to be loaded onto hemoglobin. So one thing to keep in mind for later is that oxygen and carbon dioxide do not compete for the same site on hemoglobin. So as carbon dioxide actually binds to the proteins in the hemoglobin. Recall that oxygen is binding to the iron in the heme group. So we see that carbon dioxide and oxygen are not competing for a binding site on the iron in the heme group. Only the oxygen is binding there. So oxygen release depends on the need for oxygen in the tissues that red blood cell happens to be passing through. So hemoglobin rarely, if ever, will unload all of its oxygen molecules that it's carrying. Even at tissues that are working really hard and demanding a lot of oxygen, hemoglobin will release some of its bound oxygen molecules, but it'll still remain fairly well saturated. So in determining oxygen saturation of tissues, the value of greatest interest in healthcare is the percent saturation. So that is the percentage of hemoglobin sites that are occupied by oxygen in a patient's blood. Clinically, this value is commonly referred to as percent sat or even sats. So roughly 80% of the CO2 that is produced by the tissues is converted into bicarbonate, which is then released into the bloodstream. The remaining 20% of CO2 is bound to hemoglobin and is carried to the lungs. Carbon dioxide is converted to a substance called carbonic acid inside of the erythrocyte by an enzyme called carbonic anhydrase. Carbon dioxide is also converted to carbonic acid in water, which means that the fluid, including blood plasma, that contains carbon dioxide will become acidic. Carbon dioxide is unstable, and it will very quickly lose a hydrogen or a proton and become a substance called bicarbonate. Bicarbonate is then released into the bloodstream, so that is, if it's been converted into bicarbonate within the red blood cell, that red blood cell will allow it to diffuse into the bloodstream. We see that bicarbonate is not acidic, and the conversion of CO2 to bicarbonate provides a very convenient way for the blood to carry the byproducts of CO2 without changing the blood chemistry. So additionally, bicarbonate will accept protons, that is hydrogen ions from acids in the blood, which will help to buffer the blood in the event of an increase in blood acids. So recall that acids are substances that will donate protons or hydrogen ions to other molecules. So the stockpile of bicarbonate that forms in the bloodstream is what is called the alkaline reserve. So let's take a step back for a moment and review enzymes, because we saw that there was an enzyme that was present in the erythrocyte to convert CO2 into carbonic acid. So enzymes are catalysts. These are substances that help chemical reactions proceed more easily with less direct energy input into that chemical reaction. Many chemical reactions require enzymes to proceed very easily. So without enzymes, we see that high amounts of energy or high temperatures or high pressures are needed for reactions to occur. So enzymes will function to hold substrates in alignment with one another so that the two substrates can chemically bond to one another. Other enzymes will function in breaking chemical bonds, such as the enzymes used by the digestive system, for example, breaking down starches into carbohydrates. So therefore, enzymes we see can be used to construct molecules, or they can be used to break down molecules. Different enzymes are needed for different reactions because a single enzyme cannot catalyze all the different reactions within the body. So that is, enzymes are very specific to the reactions that they catalyze. So we have thousands of different enzymes in the body. So several important aspects about enzymes are that the enzymes itself, or the enzyme itself, is not altered during the course of the reaction, and they are not used up by the reaction. So an enzyme, which is shown here in green, is typically a protein that has active sites. That is, the active sites are places where substrates can bind and be properly aligned for chemical reactions to take place. So remember that molecules, although they are very small, do have a three-dimensional shape. And so for chemical reactions to occur, we see that certain molecules have to be properly aligned so that they can form new chemical bonds, or in the examples of enzymes in the digestive system, so that chemical bonds can be broken. So in this example, two substrates bind temporarily to the enzyme and are properly aligned so that the new chemical bonds can form between them, and the two substrates can bind together, and form the product of the reaction. The product on the shown in part C and D of this particular diagram is then released by the enzyme, and the enzyme, as we see, remains unaltered so that it can catalyze another one of the same reactions. So keep in mind that enzymes can also break substances down into smaller components. This example, of course, just happens to show that an enzyme is building a product from two substrates. So just like we would see with carbonic anhydrase, building carbonic acid from carbon dioxide and water. So when bicarbonate reaches the lungs, it re-enters the red blood cell and is converted back into carbon dioxide, which is a gas, and that is then released during exhalation. This is also an enzyme-driven process, although we don't have the name of the enzyme in this particular diagram here. So about 20% of the CO2 that's produced by the tissues is carried by hemoglobin. CO2 that binds to hemoglobin actually binds to the globin proteins, so does not actually bind to the heme groups. So again, this just shows that CO2 is not competing with oxygen for the same binding site on the heme groups. The binding of CO2 to hemoglobin occurs more easily when hemoglobin is relatively deoxygenated, which is very ideal because typically CO2 concentrations are going to be the highest at the tissues where that CO2 is being produced. The tissues are also where oxygen is being unloaded from the hemoglobin so that those tissues can use oxygen in their metabolic processes.